somewhere in a hospital corridor in the state of Washington, a mother was told the one thing no mother should ever have to hear. The DNA test says these are not your children. She had carried them. She had given birth to them. Her husband had watched both deliveries with his own eyes. And still, the paper in front of her said, in the coldest language science has ever produced, that she was a stranger to her own sons and daughter. For a moment, imagine what that does to a person. Not a broken bone, not a virus, not a diagnosis with a name you can look up and prepare for. A quiet, bureaucratic sentence that seems to unmake your entire identity, using the one piece of evidence our species has agreed is beyond argument. DNA, the molecule we trust more than eyewitnesses, more than photographs, more than memory itself. And yet, here it was, apparently lying to a courtroom about a woman's own children. This is not a plot from a crime drama. Her name became public only because a lawyer refused to accept what the machines were saying, and what he eventually uncovered would crack open one of the strangest secrets biology has ever kept, one that forces us to ask a question most of us have never thought to ask. Are you actually made of only one person's DNA? Or is it possible that somewhere inside you, quietly, invisibly, there is a second genome belonging to someone who never got the chance to be born and who exists now only as a hidden thread woven into your own body? To understand how something like this could even happen, we have to go back to the moment every human life begins, in the first hours after conception, when what will become you is nothing more than a single cell dividing over and over inside your mother's body. Ordinarily, this is a clean, singular story. One sperm, one egg, one zygote, one blueprint, copied faithfully into trillions of cells that will one day become your skin, your heart, your eyes, your voice. But biology is not always clean. Occasionally, a woman releases 2 eggs instead of 1, and both are fertilized by 2 different sperm cells, carrying 2 different sets of genetic instructions. Normally, this produces 2 separate embryos, 2 individuals, fraternal twins, growing side by side, sharing a womb but never sharing a body. But in an almost unbelievably rare set of cases, something else happens first. In the earliest days of development, before either embryo has become anything resembling a person, the 2 clusters of cells find each other and, biologically speaking, a living merger of twins who were never meant to be born as 2 people. Scientists call this condition tetragametic chimerism, named for the 4 gametes, 2 eggs and 2 sperm, that combine to create it. And the woman in that hospital corridor, the one accused by science of not being her own children's mother, was one of the very first documented human chimeras ever to have her case become public. Her name is Lydia Fairchild, and for years, the state pursued her for welfare fraud, insisting that a woman who does not share DNA with her children could not possibly be entitled to support them, no matter what her body, her memory, and her family told the world. Her case might have ended in catastrophe if her lawyer had not stumbled across a nearly identical story published quietly in the New England Journal of Medicine about a woman named Karen Keegan. Karen's discovery began, almost unbelievably, with an act of love. In 2002, she needed a kidney transplant, and her 3 adult sons immediately volunteered to be tested as potential donors. What came back should have been impossible. According to the genetic tests, only one of her 3 sons was actually related to her, not 2 of 3. Not a mix-up in the lab. The math simply did not work. She'd carried and raised all 3 of these young men, and yet their DNA said she was, biologically, a stranger to 2/3 of her own family. Doctors eventually tested tissue from nearly every part of her body, and they found the answer hiding in an unlikely place, her thyroid. In that single organ, a second, entirely separate genetic signature was quietly present, one that did match her son's. Karen Keegan, like Lydia Fairchild after her, had absorbed her own twin in the womb decades earlier, and had been walking through life for over 50 years as 2 people fused into one, without a single symptom, without a single reason to ever suspect it. Once you understand what happened to Karen and Lydia, an unsettling thought starts to take shape. If chimerism can hide for 50 years inside a woman who shows no outward signs of anything unusual, how many other people might be walking around the same way, completely unaware? For a long time, doctors assumed this condition was a medical curiosity so rare it barely deserved mention in textbooks. But newer research is quietly overturning that assumption. Cases are increasingly found not through some dramatic percentage of the population. Individuals living ordinary lives, holding down jobs, raising families, could be low-level chimeras without ever knowing it. Because unless someone happens to test the right tissue, the second genome simply never surfaces. And in certain rare individuals, the evidence does not stay hidden in a lab report. It becomes visible on the skin itself. There's a woman whose case became famous not through a courtroom, but through her own willingness to speak publicly about her body, a model and musician who grew up noticing an unusual line running down the center of her torso, dividing two slightly different skin tones, like a boundary drawn directly onto her body by whatever process shaped her in the womb. For years, she had no explanation, until genetic testing confirmed what doctors suspected. She, too, was a tetragametic chimera, and the very line on her skin marked the literal border between the two genetic populations of cells that made her. In effect, she carries a visible map of her own vanished twin, drawn onto her body every single day of her life. Chimerism forces us to confront something deeply uncomfortable about the story we tell ourselves regarding identity, that each of us is one continuous, singular genetic story from conception to death. But the human body, it turns out, is far messier and more collaborative than that. And chimerism is not even the strangest way a single body can end up carrying more than one genetic story. There is a second, subtler phenomenon, one that does not require two embryos at all, called mosaicism. Instead of two separate individuals merging together, mosaicism begins with a single fertilized egg, a single genome, no twin involved whatsoever. But as that one embryo divides, again and again, a random mutation occurs in one of the dividing cells, and every single cell that descends from that one altered cell going forward will now carry the mutation, while every other cell descending from its unaffected sibling cells will not. The result is a person built not from two people, but from two versions of the same person, one carrying a genetic change, the other never inherited, distributed unevenly through the body like patches on a quilt. Mosaicism turns out to be shockingly common, more common by far than chimerism, and scientists now believe that virtually every human being who has ever lived carries at least some small degree of it, tiny clusters of cells scattered through the body with mutations that exist nowhere else in that same person, invisible variations that came from nowhere but the ordinary imperfection of cell division itself. In most cases, these differences are so minor and so isolated they never matter at all. But in rarer instances, mosaicism can explain medical mysteries that once seemed impossible, a birthmark shaped in strange geographic patterns following what is called the lines of Blaschko, one side of a person's body aging or developing subtly differently from the other, or condition appearing in only part of an organ instead of throughout it. Increasingly, forensic geneticists have had to reckon with the fact that mosaicism, like chimerism, can complicate the supposedly unshakable authority of a DNA match, because the very organ you happen to swab for a sample might carry a genetic signature that a different tissue in that same body would flatly contradict. If chimerism and mosaicism prove that a single body is not always one unbroken genetic story, then the case of a Scottish woman named Jo Cameron proves something even stranger, that a single, precise alteration in one obscure stretch of DNA can rewrite what it feels like to simply exist inside your own skin. For most of her adult life, Jo Cameron had no idea she was different from anyone else. She endured a severely degenerated hip joint that should have caused agonizing daily pain and felt nothing. She underwent hand surgery for osteoarthritis, a procedure typically followed by weeks of recovery and medication, and required none. She had spent her entire life bruising, burning, and injuring herself in the ordinary chaos of daily living, and simply never connected the dots, because to her, this was normal. It was only in her 60s, when doctors realized her lack of pain after major operations defied every expectation of human physiology, that researchers at University College London began the 6-year search that would eventually lead them to a gene nobody had ever named before, one they eventually called FAA-out. What they found rewired an entire assumption about the human genome. FAA-out sat beside another gene responsible for regulating a natural compound in the body chemically related to the active ingredients in cannabis, one involved in modulating pain, mood, and fear. And Jo Cameron, a rare mutation, had quietly dialed down this entire system, turning the volume on pain almost all the way to silence, while simultaneously suppressing anxiety and fear, but not every extraordinary genome belongs to someone still walking among us. Some of the most astonishing DNA ever studied belongs to people who have been dead for tens of thousands of years, preserved by nothing more than cold, dry luck in caves scattered across Europe and Asia. For most of human history, we assumed that Homo sapiens simply replaced every other human species that ever existed, the Neanderthals vanishing into extinction while our ancestors marched forward alone. Then, in the early 2010s, researchers successfully sequenced traces outside of Africa carry somewhere between 1 and 2% Neanderthal DNA, quietly embedded in their genome, inherited fragment by fragment from a species we once assumed had left no descendants at all. That inherited Neanderthal DNA is not simply an evolutionary souvenir sitting quietly in the background of the genome. Some of it is actively shaping human bodies today. Certain Neanderthal-derived gene variants have been linked to stronger innate immune responses, better equipping some modern humans to fight off certain infections, a legacy advantage passed down from a hardier, cold, adapted species that had already spent hundreds of thousands of years surviving in Ice Age Europe before modern humans ever arrived. Other inherited fragments have been linked, less fortunately, to increased risk for certain autoimmune conditions and altered susceptibility to viral illness, a genetic inheritance that can cut both ways, depending on the environment a person's ancestors happen to live through. And in an even stranger twist, researchers studying a different group of ancient humans, the Denisovans, known mostly from a single finger bone and a few teeth discovered in a Siberian cave, found that modern populations living at extreme high altitude in Tibet carry a Denisovan-derived gene variant that allows a separate, extinct branch of the human family tree, tens of thousands of years ago, and still quietly protecting their descendants today, every time they climb. If ancient human DNA can rewrite what we thought we knew about our origins, then some of the most resilient DNA ever discovered belongs not to a human at all, but to organisms that seem almost deliberately built to defy destruction itself. Deep within research labs, scientists have spent decades studying a bacterium so resistant to damage that it earned the nickname Conan the bacterium, capable of surviving radiation doses thousands of times higher than what would instantly kill a human being, doses so extreme they would shatter an ordinary organism's DNA into thousands of fragmented, unreadable pieces. And yet, this bacterium simply reassembles its own genome afterward, stitching the broken fragments back together with a repair. Efficiency that puts nearly every other form of life on Earth to shame. Researchers still do not fully understand how it achieves this feat, only that its DNA repair machinery appears to be operating on a level of precision most organisms never needed to evolve, because nothing on Earth's surface was ever supposed to survive that much radiation in the first place. Alongside it sit tardigrades, microscopic eight-legged creatures barely visible to the naked eye, capable of surviving being frozen to nearly absolute zero, boiled at extreme temperatures, dried out completely, physically shield their DNA from radiation damage, essentially wrapping their genetic code in molecular armor, a survival strategy so effective that researchers have begun exploring whether inserting versions of that same protective protein into human cells might one day help protect astronauts on long-duration missions to Mars from the very radiation exposure that currently makes such journeys so dangerous. And then there's the strangest case of all, DNA that was never supposed to still exist, taken without consent, from a woman who died in 1951, that has gone on to outlive nearly everyone who was ever involved in the story. Her name was Henrietta Lacks, a young mother being treated for cervical cancer at a patient consent for such things was rarely even considered. What made her cells extraordinary was not a rare mutation for pain or an ancient inheritance from a vanished virtually every other human cell line scientists had ever attempted to grow in a laboratory. Hers simply refused to die. They kept dividing a vaccine to study the effects of radiation and toxins on human cells, to research cancer treatments, and to help lay the groundwork for the COVID-19 vaccines that reached hundreds of millions of arms decades after her death. Today, there are estimated to be more of her cells living and dividing in laboratories around the world than existed in her entire body while she was alive, a strange, unintended kind of immortality, built entirely from one woman's uniquely aggressive genetic code, extracted without her ever knowing it would outlive her by more than 70 years, and counting. There is one more mutation worth pausing on because, unlike almost everything else in this story, it did not simply get discovered by undergoing treatment for both leukemia and HIV, a combination that left his doctors needing a bone marrow transplant to save his life from the cancer. But instead of simply finding the closest immunological match, his physician made an unusual decision. He went looking for a donor who also happened to carry a specific, naturally occurring mutation in a gene called CCR5. A mutation known to block the very receptor that HIV uses to enter human immune cells in the first place. It is astonishingly rare, found in only a small fraction of people of Northern European descent, the likely legacy of ancient plagues that quietly favored anyone whose immune cells simply could not be infiltrated the same way. The transplant worked on two fronts at once. The new bone marrow cured his leukemia, and along with it delivered an entire immune system built from cells that HIV could not infect. Years later, doctors confirmed what once would have sounded impossible. The virus was undetectable in his body, even after he stopped all antiretroviral medication entirely. He became known publicly as the Berlin patient, and eventually, several more patients following similar transplant protocols achieved the same result, opening an entirely new, if currently impractical, avenue of research into a functional cure for one of the most devastating viruses in modern history, built entirely around a single, naturally occurring genetic accident, one that had been quietly sitting in a small fraction of the human population for centuries, waiting for medicine to catch up to what evolution had already written. Not every protective mutation announces itself through a single dramatic case. Some hide inside entire communities, passed down for generations, only becoming visible once researchers finally think to look closely at the right isolated population. High in the mountains of southern Ecuador, there exists a small, close-knit community whose members share an extremely rare genetic condition called Laron syndrome, caused by a mutation that blunts the body's ability to respond to growth hormone, resulting in unusually short stature, often well under 5 feet tall as adults. For decades, this community was studied mainly as a curiosity of extreme growth hormone resistance. Then, in the early 2010s, an endocrinologist tracking the group for years noticed something remarkable buried in decades of medical records. Despite carrying genetic backgrounds and lifestyles that should have made them just as vulnerable to cancer and diabetes as their unaffected neighbors and relatives, individuals with Laron syndrome in this community showed a dramatic reduction in both diseases, developing tumors and diabetes at a fraction of the expected rate, even when they engaged in the same diets and habits that put others at risk. Laboratory experiments later suggested that the same hormonal pathway responsible for their small stature may also be suppressing the very cellular growth signals that allow cancerous cells to multiply unchecked, an accidental built-in brake on runaway cell division, hiding for generations in plain sight inside one small mountain community, waiting for medicine to notice. Every one of these stories, the accidental twins fused into a single body, the woman who cannot feel pain, the inherited fragments of a vanished species still adjusting our immune systems today, the bacterium that shrugs off lethal radiation, the cells that simply refuse to stop dividing, forces the same conclusion from a slightly different angle. The genome each of us carries is not a fixed, finished blueprint we simply inherit and pass along unchanged. It is closer to a living document, one written imperfectly, copied with small errors, occasionally merged with someone else's draft entirely, sometimes borrowed from an entirely different species thousands of generations removed, and now, for the first time in the history of life on this planet, capable of being deliberately edited by the very organism whose code it represents. We are the only species that has ever looked directly at its own instruction manual, understood enough of the language to begin correcting the typos, and started asking, seriously, urgently, and without a clear answer, yet how much of that manual we actually have the right to rewrite. The people at the center of these stories, Lydia Fairchild fighting to keep her own children in a courtroom that trusted a lab report over her own memory, Karen Keegan, discovering an invisible twin she had carried inside her for half a century, Jo Cameron learning in her 60s that a single hidden mutation had quietly spared her a lifetime of suffering. Most people simply endure, never set out to become case studies. They were simply living their lives until one unexpected medical crisis pulled back the curtain on something extraordinary that had been sitting inside them the entire time, unnoticed, unremarkable, until it wasn't. And that, in the end, might be the most humbling revelation of all. Somewhere inside your own cells right now, there could be a mosaic patch of mutation nobody has ever tested for, an inherited fragment from a species that vanished tens of thousands of years ago, or simply a story more complicated than the single, tidy narrative we all assume our own DNA is quietly telling. We spend so much time looking to the stars for mystery, when the strangest, most unresolved frontier in all of biology might be sitting inside every one of us, 3 billion letters long, mostly unread, and still, after all these years of discovery, only just beginning to be understood.